Hello, and welcome to the Terra 201 tutorial, demonstrating the use of Terra for viral genomics. This tutorial will cover importing data from NCBI's SRA into our COVID-19 workspace, running workflows with the data, and using Jupyter Notebooks to run a quality check on the data. We'll begin with a short overview of how Terra works on the cloud. Terra is built to streamline the interaction of three types of scientists: data generators, such as clinical scientists; tool developers, such as the computer scientists here at Broad; and researchers, laboratory scientists who want to turn data into actionable observations. A Terra workspace is a computational sandbox that lives in the cloud and can access local and cloud-based resources outside of itself, as well as store data in its own dedicated Google bucket. Here is a simplified illustration that highlights the concept of integrating access to data into the user interface. If you use Terra, you don't have to navigate your data using the command line or almost any code. The workspace is already wired up in such a way that your workflows and notebooks can access various data libraries using the data model. The workspace is a portal that allows you to interact with data and Google Cloud resources, which you can requisition on a flexible basis. One aspect of this flexibility is a result of the fact that Terra includes underlying architecture that supports most cloud vendors, although this tutorial runs specifically on the Google Cloud. In this tutorial, we'll be focusing on our COVID-19 workspace, which contains COVID-19 genomic data and workflows that enable you to perform a variety of genomic analyses. This tutorial will be focusing on importing data directly from NCBI's SRA, given an SRA ID, running assembly, and doing some quality checks to the FASTA files. We'll begin the tutorial by going to Terra's landing page. Go to the URL app.terra.bio and click on the main menu button in the top left corner. Then click on Library, and then click on Showcase. This is the Terra Library's Showcase and Tutorial section, where you'll find a variety of workspaces designed to help you acclimate to working with Terra. In the left column are new and interesting workspaces we've recently published on various relevant topics. The middle column contains featured workspaces meant as tutorials for using Terra. And in the rightmost column, you can see workspaces that contain our GATK workflows preset to their best practices configurations. The workspace we'll be using in today's tutorial is called COVID-19 and is in the middle column, about halfway down. Go ahead and click on this workspace. Once you click on the workspace card, you'll be taken to this view. You are now looking at the public version of the workspace. You'll notice that you aren't listed as one of the owners of the workspace on the right-hand side, and also that this is a read-only copy. In order to be able to access any computational resources, you'll need to clone your own copy of this workspace attached to a billing project that you have access to. Do this by clicking the three-dot button at the top right of the screen, and then selecting the clone option. Give your workspace copy a unique name, and choose a billing project that you have access to. Once you click Clone Workspace, you'll be automatically taken to your own clone of this workspace. As you can see, you're now listed as the owner of this workspace. Let's quickly go over the contents of each workspace. The first tab in the workspace is called the Dashboard. The dashboard contains an overview of the contents of the workspace, and you'll often find cost estimates and expected runtimes for workflows here as well. This includes the workflows Fetch SRA to BAM and Assembly Ref-based. In this dashboard, we also provide background information on what data should be run as an example when you're just getting started. Next to the Dashboard tab is the Data tab. This is where you can access the workspace's data model. The workspace data model is Terra's system for organizing data and metadata in such a way that allows your workflows and notebooks to interact with the data you wish. In this sample table, you'll find the metadata that was available publicly on SRA, associated with each one of these SRA IDs, as well as the BAM files associated with each sample. Under the Data tab, you can also find other workspace-related data, such as reference files, and you can also access your workspace-dedicated Google bucket, where you'll see the results of some of your workflows populate, depending on how your workflows are configured. Next is the Notebook tab, where the workspace organizes your Jupyter notebooks. You can upload notebook files here or create your own notebook from scratch. Terra supports Jupyter notebooks in R or Python. Under the Workflows tab, you'll find all of your workflows listed. This workspace has quite a few workflows, 2 of which we'll launch as part of this tutorial. Once we launch those workflows, they'll appear in the Job History tab, which is currently empty. Here you can track the progress of your workflows and relaunch any that fail. Let's try launching some workflows so that you can get a feel for how this works in practice. You can launch workflows by either selecting the data first or selecting the workflow first. We'll do it both ways in this tutorial. Let's start by returning to the Data tab. To see how we can fill this table out using our workflows, let's start by deselecting some of the metadata columns by clicking on this icon, selecting None, and then selecting just the accession ID, so that we're left only with our sample ID and accession number. This will make it easier to see what data we'll be adding with the workflow we'll now launch. For this demonstration, let's just select the top 3 rows and select Open With. Next, you'll select Workflows, and from the list of available workflows, you'll select Fetch SRA to BAM. This workflow will fetch the BAM file and any metadata that was associated with the BAM file from NCBI's SRA. Make sure that you've selected the master version of the workflow. Before we launch this workflow, we'll have to do a couple of things. We've already selected which data we want to run, but we need to fill out some of these configuration attributes. Under Inputs, we need to make sure that the workflow knows to look for the number in the column labeled accession in the data table. The syntax for pointing at cells in a sample data table is this dot and then the name of the column. Fill in this dot accession into the SRA ID input variable. We're almost ready to launch this workflow. But before we do, we need to make sure that our outputs are also configured. If you click on the Outputs tab in this workflow, you'll notice that a lot of the required attributes are unfilled. Since we want to be sending metadata and links back to our workspace data model, we'll have to use a similar syntax. Fortunately, there's an easy shortcut for filling them all out. You can just click Use Defaults. This will fill out all of the attributes with the correct syntax, so that upon completing, the workflow will create columns based on the variable names given in the variable column, and populate the cells in those columns with metadata and links associated with those SRA IDs. Now click Save, and you'll be ready to launch your workflow. Once this workflow completes, you'll be able to see under the Data tab that our data table has a new column, with the 3 rows we selected containing a new entry for the BAMs that we've fetched. Now that we've imported our BAM files into our workspace, we could go ahead and run our reference-based assembly workflow using the reference FASTA we have in our workspace data. This workflow will run the assembly and output FASTA files for each sample, along with the associated assembly metrics, such as assembly length. Since we are running the workflow using our data model, the outputs will get appended to our sample table, providing for a helpful and comprehensive data table that we can either download as a TSV or read into a Jupyter notebook for generating QC plots. Let's try launching one more workflow to see another way to select data against which to run your workflows. We'll go back to our Workflows tab, and this time we'll select Assemble Ref Based. Again, we'll make sure that we're using the master version of the workflow, and this time we'll select the data that we want to run from this view. Instead of selecting from our sample table, we'll select from our sample set table. After you select sample set from the drop-down menu, click on the Select Data button and select the bottommost row. We do have to modify one of our input attributes because the syntax for pointing at the sample set table is slightly different from the syntax for pointing at the sample table. To use the sample set, the correct syntax is this.samples. The name of the column. The sample set is composed of samples from the sample data table, so you'll notice that if you delete your entire sample data table, you will also delete the sample sets that are composed of entries from the sample table. The next step after you've successfully assembled your genomes is to look at some metrics, such as assembly length, in order to assess if your data look good enough for downstream analysis. To do this, we'll use the Jupyter Notebook contained in this workspace. To select the notebook you want to run, go to the Notebooks tab and select the notebook. You'll notice that when you first click on the notebook, you only see a read-only preview. In order to actually run the cells, you'll need to spin up a cluster. To launch a Jupyter Notebook in edit mode, you must first launch a Cloud Compute cluster using the Cloud Environment button in the top right of your screen. These computational environments can be customized to a variety of needs, but this tutorial uses the default environment, so all you'll have to do is press play. It may take a few minutes for this process to complete. Once your cluster is running, you'll be able to open this notebook in edit mode, which allows you to edit the contents of the cell and to run each cell on your virtual machine. Once the notebook is ready to go, you can run each cell one by one. There are a number of ways of running the cells in the Jupyter Notebook. For example, by using some of the options in the Cell drop-down menu. However, I'll be using a shortcut by simply pressing Shift-Return on each cell that I've selected. We'll start by running the very first code cell, which imports packages that are necessary to run the rest of this notebook. In the next cell, we'll set up our workspace environment variables, which is how we make sure that our notebook knows how to find the data in this workspace. As a sanity check, we print out some of the credentials associated with the workspace. Next, we'll load our output data and print out the head of the table in order to show that we've done this successfully. You'll notice that this is the same data as in the sample table under our Data tab. Now all that's left is to plot the data. We'll start by importing the necessary packages to do the plotting, and then creating a data frame that includes the mean coverage and the assembly length unambiguous for our samples. Creating simple bar plots of this data will allow us to quickly visualize if our genomes could be fully assembled. By generating these plots, we can clearly see that not all of our samples are fully assembled, or have less than 98% of the desired nucleotide length. From here, we can quickly discard those samples for downstream analysis. This concludes our demonstration of using the COVID-19 workspace. For more information, you can visit our knowledge base, where you can find detailed documentation for this workspace, as well as a wealth of information on how to use Terra. Thank you for listening.